My name is Joshua Mugabi. I welcome you to Topic 1, Cell Biology. Our learning outcome C. Analyze the ultrastructure of animal/plant cells, bacterial cells, and the plasma membrane to distinguish prokaryotic and eukaryotic cell characteristics. We are looking at learning activity C. Learners search for the differences in ultrastructure of prokaryotes, design an annotated flow diagram, and use it to explain the division of the original kingdom Monera into kingdom Eubacteria and Archaebacteria. Division of Monera into Eubacteria and Archaebacteria. Item 1. We shall look at reclassification of prokaryotes. Scientists studying electron micrographs of prokaryotic cells discover that some bacteria-like organisms live in extremely hot springs and salt lakes and have cell structures that differ from common bacteria. This evidence leads to the division of the former kingdom Monera into Eubacteria and Archaebacteria. Task. State the original kingdom that included all prokaryotes. Name the two kingdoms formed after reclassification. State 3 ultrastructure differences between Eubacteria and Archaebacteria. Explain why these differences justify placing them in separate kingdoms. State the original kingdom that included all prokaryotes. The kingdom is Monera. Name the two kingdoms formed after reclassification. Eubacteria and Archaebacteria. State 3 ultrastructure differences between Eubacteria and Archaebacteria. We have the features: Eubacteria, Archaebacteria. The cell wall. Eubacteria have peptidoglycan present. Peptidoglycan is absent in Archaebacteria. Cell membrane lipids. They are ester-linked lipids. In Archaebacteria, they are ether-linked lipids. The ribosomes. Eubacteria have got 70S bacteriotype. Archaebacteria 70S but structurally different. DNA. Eubacteria has no histones. Archaebacteria, histone-like proteins are present. Flagella. Eubacteria has a simple flagella. Archaebacteria, structurally different Archaebacteria. Explain why these differences justify placing them in separate kingdoms. Differences in cell wall composition, membrane lipids, and genetic organization indicate fundamental structure and biochemical differences. These differences affect metabolism and survival strategies, justifying classification into separate kingdoms. Division of Monera into Eubacteria and Archaebacteria. Item 2. We are going to look at cell wall investigation. A student tests the effect of penicillin on two prokaryotic organisms. One organism is killed, while the other is unaffected. Task: identify the type of prokaryote affected by penicillin. Explain why the other organism is not affected. Relate this observation to ultrastructure differences. Identify the type of prokaryote affected by penicillin. Eubacteria. Explain why the other organism is not affected. Archaebacteria lacks peptidoglycan in their cell walls. Relate this observation to ultrastructure differences. Penicillin inhibits peptidoglycan synthesis. Absence of peptidoglycan in Archaebacteria makes them resistant. Division of Monera into Eubacteria and Archaebacteria. Item 3. We are looking at extreme environments. Organisms are discovered thriving in acidic hot springs and saltwater lakes. Task: identify the kingdom to which these organisms belong. State 2 ultrastructure adaptation that enable them survive. Explain how these adaptations support the reclassification. Identify the kingdom to which these organisms belong. Archaebacteria. State 2 ultrastructure adaptation that enable survival. Ether-linked membrane lipids resistant to heat. Unique cell wall composition. Explain how these adaptations support the reclassification. These structures provide stability in extreme conditions. Such unique ultrastructure supports separation from eubacteria. Division of Monera into Eubacteria and Archaebacteria, item 4. Let's look now on genetic and ribosome evidence. Researchers find that the ribosomal RNA and transcription machinery of some prokaryotes resemble those of eukaryotes. Task: identify the group showing this similarity. State one structure, ultrastructure, and molecular feature supporting this. Explain its taxonomic significance. Identify the group showing this similarity. Archaebacteria. State one ultrastructure or molecular feature supporting this. Presence of histone-like proteins, unique RNA polymerase. Explain its taxonomic significance. Indicates evolutionary divergence and supports classification into a distinct kingdom. Marking emphasis. State correct kingdoms. Compare ultrastructure features. Use correct biological terminologies. Explain classification logically. Apply knowledge to real examples. Conclusion: ultrastructure differences in cell walls, membranes, and genetic material justified the division of the former kingdom Monera into Eubacteria and Archaebacteria. Thank you for participating in Topic 1 Cell Biology, Learning Outcome C. Analyze the ultrastructure of animal, plant cells, bacterial cells, and plasma membrane to distinguish prokaryotic and eukaryotic cell characteristics. Our learning activity has been C. Learners search for the differences in ultrastructure of prokaryotes. Design an annotated flow diagram and use it to explain the division of the original kingdom Monera to kingdom Eubacteria and Archaebacteria. If it has been useful to you, visit me at Mugabi Biology on YouTube.